Hi, so today we're going to talk about DNA transcription and translation, which really equates to a gene being expressed. This comes from chapter 12. We've already covered parts of chapter 12 where we reminded ourselves what DNA is. And so this is the rest of chapter 12 that really covers like when a gene makes a product, it's a protein, and we say that gene is being expressed. So a gene is essentially like a trait. It's a hair color or it's anything that can be considered a trait. Molecularly, it's really a length of DNA, and that DNA has a sequence of nucleotides that code for a specific protein. An allele is a term that means it's still coding for the protein, but a slightly different one. So the sequences in that nucleotide are just slightly different in that DNA. So you're looking at like red hair versus not red hair. So proteins are made up of one or more subunits called polypeptides. This is not new information. We learned about this information way back in chapter 3 when we talked about proteins. So one gene essentially codes for one polypeptide, which is a chain of linked amino acids. So generally speaking, another way to say that is DNA actually gets transcribed into mRNA, and you're still talking the same language, just different forms of the language. So in English, it'd be spoken English versus written English. In cells, it's DNA versus RNA. And then RNA gets translated into a protein where initially you were talking about nucleic acids, now you're talking about amino acids. The process of transcription and translation is pretty straightforward. So transcription is the first step in making proteins. So transcription and translation as a topic is all about making some proteins. So it's basically the process of taking that DNA or that gene that DNA has and converting it into an mRNA strand. Remember, we said that has to happen because DNA can't leave the nucleus, and we know that ribosomes are where proteins are made. So genes have three distinct regions. There's an initiation sequence where an enzyme that starts transcription attaches. There's an RNA coding region where the actual code for the protein exists. And then there's a termination region that basically tells the enzyme, hey, you're done with this process. So first, the enzyme attaches to the initiation sequence and unwinds the DNA. Remember, DNA is double-stranded and it's wound, right? Only one of those strands has the sequence for the gene, and that's called the template strand. The other strand is called the nonsense strand. So, essentially, an mRNA copy is made from the DNA template strand using the enzyme RNA polymerase. So, this image is actually a cute view of basically looking at how the DNA is attached to the RNA polymerase enzyme, and that enzyme helps read this code. So next, an mRNA copy is essentially made until the RNA polymerase reaches that termination region, and that causes the enzyme to fall off, and then DNA rejoins. Here's another image of transcription in the nucleus. Here's transcription of DNA to RNA. So you guys have the task of transcribing the DNA to RNA. And now remember, transcription from DNA to RNA requires you to think about the fact that RNA has uracil instead of thymine, okay? So if I were to do this, and I would definitely pay attention to this task because this is something that we do on our next lecture exam. So if I give you a template strand, I expect that you might be able to kind of match it up and tell me what letters would go here. So C here, G, G, A. Remember, this is RNA right here. So this means this would have to be a U. My little writing thing isn't awesome, but it works. So this is A, and then this is U, G, C, U, A, G, C, U, and A. So I would expect you to be able to do this, of course, a little bit more neatly on an exam. Translation is essentially taking the code that DNA gave RNA and then translating it into the protein code of amino acids. So, just about proteins. Proteins are made up of amino acids, and they're polymers of amino acids. There are only 20 different amino acids present, but there are hundreds of thousands of different proteins. So really, how can that be? How can there only be 20 amino acids, but there's hundreds of thousands of different proteins? Essentially, it's comparable to the English language. So when you think about it, how many letters are in the alphabet? Only 26. But how many words are there? Really, there's a ton. You could go on and on. It's almost infinite on how many words there are, and there are always more being made. Not good ones, but there are more being made. So every word has a unique structure of letters using those 26 letters that you have, and that's exactly like proteins and amino acids. You have 20 different amino acids, but combining those in different arrangements gives you so many protein options. So proteins are made up of those 20 different amino acids bonded together to form polypeptides. So once the mRNA leaves the nucleus, it goes to a ribosome in the cytoplasm, and together with transfer RNA will then produce proteins. So first, RNA leaves the nucleus, binds to a ribosome. Only 6 RNA nucleotides are in the ribosome at one time. So the ribosome will look for what's called a start codon. This is very critical that you know that in this process there's a start codon. And a codon is just a group of 3 nucleotides on the messenger RNA that specifies for a specific amino acid. So there are 64 codons. So the ribosome will look for a start codon. So if you are going to pretend that you were doing this job, if you were doing translation on my little paper here, you would take this bottom line here, like you would take this bottom line where it says RNA, and this RNA would be translated. And so you wouldn't start the process of translation until you found the combination of letters A, U, G. So here is our start codon right there. So to understand what AUG means, there's actually a chart that helps you understand and helps you know which amino acids RNA is coding for. So this is super cool. So if the RNA codon is AUG, you find A here, right? And then you find U here, and then on the right side you find G in that A column. And so here, and then here, all leads to the fact that AUG is coding for met or methionine. So then if I challenged you to find what U coded for, so you come up here to U, right here, then you find U here, and then you find U here. So if you go here, here, it's almost like, it's really fun. Okay, so then this PHE, AGA, so you go start an A here, and you go over to G, right here in this column, and then A, and you can see that AGA codes for ARG. So we have some special codons, like the AUG codon is a start codon. So I always say, just like school starts in August, proteins start with AUG. And so AUG actually codes for the amino acid methionine. And there are also 3 stop codons. So those stop codons actually tell the ribosome to release the mRNA. So if we go forward, we'll find those stop codons. So far, mRNA has left the nucleus, attached to a ribosome. The ribosome found the start codon. Then an amino acid is attached to a transfer RNA molecule and brought over to the mRNA. This tRNA molecule has a sequence that pairs up, makes complementary base pairs with the codon in the mRNA strand. So if the first codon, if the first tRNA codon that's going to be brought up, it's going to be something that makes a complementary base pair with AUG, right? So it would be UAC. So the tRNA binds to the mRNA sequence, adds the amino acid, tRNA leaves, and those amino acids bond together. Essentially, this image shows a ribosome reading the mRNA, and for each codon that it reads, it forms a correct base pair, and then the amino acid bonds, and once it's bonded, the tRNA leaves. And that actually continues until a stop codon is reached, and then the amino acids disconnect from the mRNA sequence and a protein is formed. So this is like the whole transcription and translation in one image, so it's very cool. You can see what's happening in the nucleus and then out here in the cytoplasm. So if we wanted to practice with the genetic code chart, we would find the amino acid sequence for this strand. So it starts with AUG. So I'm going to write these down so we can remember. AUG, that's where we start always. That's the start codon. CGA, CGA, AUUU, AA. So we're pretending that we're ready to translate using that chart. And we'll also practice finding these codons, AUG, GGA, GAG, CAA. Okay, so if we're going to go by the first group of codons, which is AUG, of course we find A, we find U, so A, then U, then G, so we know we're looking at MET here. And then the next one is CGA, so we'd find C, G, and then we'd find A, so we know it's ARG. And then we had another CGA, so we got another code for ARG, and then CAA, so CAA, and this one is glycine. And we could even look at, we know AUG is MET, GGA, so we find G, we find G, and then we find A, so that's glycine, or C, A. So then I would want you to put the whole concept together. So if this is your DNA template strand, you would start with C, G, T, A, C, G, C, T, C, 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 T, A, G, A, C, T, A. And so I would expect you to do something similar, okay? Then you would just do complementary base pairings. So if this is DNA, right? This is our DNA. So then you would make an RNA base pairs with this. So C to G, G to C, T to A, and then A to U, G to C. Looks like our start codon is right here. C, G, A, G, 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 A, U, U, oops, A, C, U, G, A, U. So I just made complementary base pairs, and now I know I can start reading codons because I found my start codon. Did you see the AUG? So then that's MET, and then we got CGA, GGG, AUC, UGA. So then we would use our chart to find them again, and I'll let you guys do this practice problem. Cells can make mistakes though. So like in this particular scenario, if there's any kind of mistake in this code, in this process right here, if this doesn't get translated right, then you're not going to code for the correct protein. And that's a big deal. That if there's a mistake in the DNA, then you see mutations. Sometimes it's going to be randomly. Sometimes there can be external things that causes mutations. But a permanent change in a cell's DNA is a mutation. So there's point mutation, where one nucleotide gets substituted for the other. There's insertion, where you can have one or more nucleotides added to the sequence. There's deletion, where one or more nucleotides are lost. So it can be random chance, or it can be mutagen. So there are lots of things that you can expose yourself to, like UV light, X-rays, those types of things that can cause mutations. Now, somatic cell mutations are not passed from one generation to the other. So if skin cancer is not passed on, but if a mutation happens on the sex cell, those sperm and eggs do get passed on, and the offspring can be affected.